Okay, someone should lead us in a prayer. Awani Darius, can you lead us in a prayer? One has gone off. Who can lead us in a prayer very fast? Yes, Letitia. Yeah.
Good morning, teacher.
Good morning to you, Letitia. Please lead us in a prayer.
Let's humble ourselves for
Mm.
our dear Heavenly Father. Thank you for this day. We thank you for this daily blessing. We thank you for our truth. We thank you for all the students and all the staff members of Edify. Lord Jesus, throughout this hour, we going to study. We pray that you open our minds to understand. Let wisdom flow, O Lord Jesus. May we understand and get each and everything we are taught in Jesus' name we are praying.
Amen. Amen. Thank you very much, Letitia. Hey, thank you very much. Okay, let me allow you guys to freely unmute yourselves in case I've asked you a question. Okay. I think we can start now. Last week, if you still remember, we looked at okay. We looked at how materials are translocated. We're able to look at the structure of the phloem. Then we looked at how the phloem is adapted to its function. I was able to even show you some questions that have been examined before. As far as paper 2 is concerned, in UNEB, one was in 2002, another in 2010. Then we looked at several experiments which were conducted to actually prove that translocation occurs in the phloem. And then we're able to go through that graph and some of these. Now today, we're going to expound on what we call mechanisms of translocation. Now, the different mechanisms, we shall look at basically 4 different types of mechanisms, but for today, specifically, we are going to look at the mass flow hypothesis. The mass flow hypothesis, or some people call it the pressure flow, or some books will call it the Munch's or Munch's hypothesis. Munch's hypothesis. So if you have seen something like that, is what we are going to discuss today, Munch's hypothesis or the mass flow hypothesis. Now, for some of you who may not be knowing, let me inform you a little bit. In science, we have a word which is hypothesis, but sometimes we also have a word which is called a theory. And I think in everyday talk, you've heard of people using that word. That's just a theory. That's just a theory. Now, I want to school you somehow today. When we talk of a theory in science, it means it is something which has evidence. It has been proved with experiment and evidence. That's when they can call it a theory, not something like just to guess. But for a hypothesis is like an academic guess. You can say, for example, whenever I touch a plant, it produces food. Now, that is a hypothesis. It is just an academic guess. And for it to become a theory, from hypothesis to become a theory, you must carry out experiments to prove it beyond reasonable doubt so that they can call it a theory. Now, there are different hypotheses that try to explain how materials are translocated in a plant. And these hypotheses, they have evidences, but of course, they also have criticisms. They have criticisms, that's why they still call them hypotheses, because of the enormous criticisms that they have. So How do materials move once in the sieve tubes? If you still remember the structure of the phloem, when I talk of the sieve tube element, you should be with me at this moment. Now, however, different hypotheses have been suggested to explain the transport of soluble products within the sieve tubes. And these are the mass flow hypothesis, the cytoplasmic streaming, electrosmosis, and surface spreading. And for today's lesson, we are basing it mainly on the mass flow hypothesis. Now, looking at the mass flow hypothesis, or which you can call Munch's hypothesis, it is the passive movement of materials through the vascular system of the plant. And it has been suggested, that is by the scientist, and the lead scientist was Munch, that there may be a passive mass flow of solids through the phloem as a result of tag-up pressure gradient. Okay, I want you to take note of that word, tag-up pressure gradient. That is highest in the leaves where sugars are formed, and they are calling it the source, and lowest in the roots, that is the sink. Now, if you can look at this plant here, which this guy was looking at, it is having leaves, it has the stems and the roots. Now, it should be noted that in the leaves, this is where food is made, or we can call it the sugars are made. So this is called the source of the sugars. So this is the source, and once they have been made, they are translocated through the phloem down to the roots, because they are needed in the roots. The root cells need them to carry out respiration, and sometimes food is deposited there. So that's where the sink is, in the roots. That's where we have the sink. So we need to take note of that as we are discussing this. Now we're going to use this picture to try and zoom inside the phloem, the xylem, and the mesophyll cells of the leaves, because these are in close proximity to one another. So this picture here, this is representing the xylem, this tube. This other tube is representing the phloem. Now, in between the phloem and the xylem are mesophyll cells. So this is representing the mesophyll cells. I think you still have the structure of the internal structure of the leaf. All these ones are in close proximity. Okay. Now you find that in summary, this is what we are going to go through. In summary, that in the leaves, this is where we find the mesophyll cells. There is manufacturing of the sugars. Now, once sugars are manufactured in these mesophyll cells, it's going to lower the water potential here in the mesophyll cells. Once the water potential is lowered, water is going to cross from the xylem by osmosis to enter these mesophyll cells. Now, once water overaccumulates in these mesophyll cells, tag-up pressure is going to be built up. These cells are going to have a lot of water in them. It's going to build up, which in turn which sugars will be taken into the phloem, still the phloem cells here. So water is just going to be moving by osmosis, and then it's going to be flowing downward through the phloem. Now, at the same time, thinking about food that is being made in the leaves, which I'm calling at the source. At the same time, at the roots, the sugars are being utilized in respiration, and some are being converted into starch, which means that when you are having tag-up pressure building up here due to overaccumulation of water, and instead here, there is not enough tag-up pressure. That's what is bringing the pressure potential gradient, where it is highest at the source and lowest at the sink. So because of the pressure gradient, the sugars are going to be translocated alongside water via the pressure gradient that has been generated between the source and the sink, and then fluids are going to move down. So in summary, that's what we are going to be looking at. Now in the leaves, the source, which you can call the source, rapid photosynthesis occurs and sucrose accumulates in the photosynthetic cells. Now, once it accumulates, the water potential in these cells is going to be lowered, resulting into water from the xylem to enter these cells, as I've said. This causes an increase in their target pressure, where? At the source. Now, in the roots, sucrose is either utilized as a respiratory substrate, that is, or broken down to provide energy, or converted into starch for storage. Now, the sucrose content in these cells is lowered and the water potential in these cells are high. So water is going to move out. You see, water is going to move out and their target pressure is consequently going to be lowered. Therefore, a target pressure gradient exists between the source in the leaves and the sink. That's the roots and the surrounding tissues. And you can see that water can move out into the xylem, this side, or to these other surrounding cells within. So that is what is creating. This cell is a little bit going to be plasmolyzed. Like target is going to be high at the source and it's going to be low at the sink. So it's what's going to drive these, or what we are calling translocation, down to the sink. Now the source and sink are linked by the phloem, and as a result, the liquid flows from the leaves to their tissues along the sieve tube elements. It is suggested that the mass flow is aided by at the sieve plates by electroosmosis, which is the passage of water across a charged membrane. Now, electroosmosis is another kind of hypothesis which was suggested to explain how molecules, how substances move through the phloem, and I will have to talk much about it. But today we are basically looking at the mass flow hypothesis. Now, this mass flow hypothesis, as demonstrated by Munch had to carry out an experiment to prove what he was saying about what happens in the phloem, in the plants between the source and the sink. And how did he set up his model? Now, this is his model. I tried to get about 3 pictures explaining Munch's model. Here you are having, you could have, let's say, a beaker which is connected to the other beaker. And here you're going to have water molecules inside here. Now you get a flask which has a semi-permeable membrane. And in this flask, if I can call it flask A, it will have, you can put there sugars, a high concentration of sugars. And then you connect it via this connecting tube to another one with a very dilute, solution. Remember also this flask B with a very dilute solution, but its membrane is also semi-permeable. It is also in water. Now, what do you expect? Now you find that water will move from this bucket, if I can call it a bucket, to enter this flask, because by the process of osmosis, leading to building up of pressures here, which pressures are going to push these fluids through this pipe to come and then be poured in this flask B. I hope you are getting it. Now, that can actually keep on working and working until when the concentration in A and B is the same. But of course, this thing is never realized. So if you want to ensure a continuous flow or bulk flow of water that is carrying sugar, it means that you have to keep on adding sugars here. Maybe you could connect some kind of a funnel, so that you keep on adding sugar I'm assuming this is your spoon. So you keep on adding sugar, you keep on adding sugar here. And then at the same time, you have to keep on removing some certain fluids from here to ensure a continuous flow. Because in a real plant, photosynthesis usually occurs, that is, sugars are constantly added. And as well, respiration occurs at the sink. You see that? Now, going through these words, in B, contain water with dilute sugar solution. Water moves out from a higher water potential region by hydrostatic pressure gradient produced and redistributed through the connecting tube. Now, between the two containers, this is the one here. So mass flow continues until the concentration of the sugar solution in A and B are equal. And in nature, equilibrium is not reached because solutes are constantly synthesized at the source and utilized at the sink. Okay, and you can see in the above model, two semi-permeable reservoirs labeled A and B are joined by a tube. Tube A contains a concentrated sugar solution. B contains a more dilute sugar solution. The two reservoirs are immediately immersed in water containing beakers as shown above. Now, this is the setup which Munch put forth to demonstrate mass flow hypothesis. Now, if you are a person who likes reading functional approach, I got this picture from Advanced Biology, a functional approach by MBV Roberts. This is where I got this picture. And this is how they put it, that cell A has a membrane permeable only to water. It contains a solution with a high concentration of sucrose. It represents the leaf of the plant. Okay. Now, water enters cell A by osmosis because it has a lower water potential than the water surrounding it. And then here in cell B has a membrane permeable only again to water. It contains a solution with a little sucrose. With little sucrose, it represents respiring or storage regions of the plant. And then water leaves because it is forced out due to the high pressure. So here, pressure is generated here, first of all, in tube A, as a result of water entering here by osmosis, pushing the fluids in a bulk movement, okay? So they're going to flow at a higher rate because they are being pushed from here due to pressure generated here. And then they come here at a very high pressure, forcing some water to move out, you see? Now this water still can go and connect, so direction of water, so it doesn't pour out so it just connects again like that, like that. If you have connected. If you have read through Advanced Biology by Simpkins, they have the same setup like in MBV Roberts, but the difference is in Simpkins, in tube B, he put there a solution. He added there actually starch, because remember starch is osmotically inactive, so he added here starch, which has nothing. It won't affect much of the water potential here. So I think you can get the different version. So water is drawn into A by osmosis and the solution flows from A to B. Water is forced out of B by hydrostatic pressure developed here as a result of water entering from the beaker to enter by osmosis here. So the flow stops when the concentration of A and B is the same, but this never happens in nature. And I think if you have realized, I'm still explaining the same thing, but in different dimensions, so that you can be able to understand. Many people may not understand this. Now, in the living plant, it is observed that the sieve tube are lined by selectively permeable membrane, so that when the sugar concentration is high, water will be drawn into by osmosis into the leaves. In the plant, the continuous flow of water is maintained by the continued active secretion of sugars into the sieve tubes at the source and its removal at the sink. Okay. And in the above model, the water which is forced out of B returns to A via the tube, this tube T here, connecting the two beakers. In plants, the water is returned from the sink to the source via the xylem. Now, this is the picture which we are talking about. If you can now relate, that this is the xylem, and this is the phloem. And these are the photosynthetic cells that make the food or the sugars. The moment they have made the sugars will actively be taken into the phloem. Once they're in the phloem, water is going to move by osmosis into the phloem, creating a hydrostatic pressure, which is going to bring about the bulk flow of the sugars to the sink. Because at the sink, already water is moving out, and already the sugars have been utilized here, so it's less concentrated here. They have been utilized in respiration, and some has been converted into starch. So once the fluids come and build up here, some of that water is going to be pushed out into the xylem still, which will move up again. So the cycle will keep on continuing and continuing. Okay. So I hope by that you've been able to understand what I call the mass flow hypothesis. But now this mass flow hypothesis has weaknesses. Because yes, Monchi was trying to show that that's how the sugars are transported. That's the mechanism, which he called the mass flow hypothesis. But people were able to get criticisms. There are some things that his setup wouldn't explain well. What are they? Number one, it does not explain the bidirectional movement of sugars and amino acids within the phloem. Because when you observe the phloem very well, in reality, you find that the sugars move in opposite directions. They can move from up, down, or down, up, or from the leaves to, for example, if you're having, I'm assuming this is the leaf here. Okay. So we'd expect, let's say, the sugars to only be moving, let's say, from the leaf going this side. But sometimes you can find when there are some sugars which are moving this side. That is the bidirectional movement. So it doesn't explain it, because if you're looking at fluids moving due to a difference in pressure gradient, that is at the source where you have higher pressures and the sink where you have low pressures, then we wouldn't expect sugars to be moving in two different directions. But of course, in reality, they move in opposite directions. So that was weakness number one. Weakness number two, or criticism number two, it failed to explain how the soluble materials would move when the gradient of the target pressure which exists is insufficient. Because it is also observed that at times, the pressure is not there, it is insufficient. So if the pressure is not sufficient, then how come materials still move? How come materials still move? So it failed to explain how the soluble materials would move when the gradient of target pressure which exists is insufficient to overcome the resistance imposed by the sieve plates. Because the sieve plates have sieve powers, which means if the sieve tube elements have end walls, so there is some kind of pressure, so resistance that is placed as the fluids try to go through the pores. But however, electrosmosis tries to explain that phenomenon. I shall talk about electrosmosis in due time. But also, there are some evidences which try to support the mass flow, much as it has weaknesses, but it has its own evidence that's supportive. Evidence one, there is a flow of solution from phloem when it is cut or pierced by a stylet of an aphid. Okay. Yeah, so when there is a flow of solution, I think this is obvious. Then evidence number two, sucrose concentration is usually higher in the leaves and lower in the roots. And this is very true according to Munch's hypothesis and demonstration. We find that in the leaves, you're going to have higher sucrose concentration, and in the roots, we are going to have lower sucrose concentration. That is another evidence. Then another one is that mass flow was observed in microscopic sections of living sieve elements. Okay, that is another evidence. Then also evidence number 4, viruses, all growth chemicals applied to leaves are only translocated downwards to the roots. When the leaf is well illuminated and therefore photosynthesizing. So when applied to shaded leaves, no downward translocation occurs. Shaded leaves, we mean like if you get leaves, I'm assuming this is a tree with its leaves. Okay. Now there may be some leaves that are not in direct contact with the sun, which are shaded by maybe branches or other leaves, and they are shaded, or by other trees. So you find that once chemicals or viruses are applied to these other normal leaves, we find that the translocation is going to move up to the roots. When the leaf is illuminated, that is, it is carrying out photosynthesis, but it will not, they will not move down, or they will not be translocated in case some of the leaves are, those in the leaves whereby they are shaded. So all those ones were critical observations to support the hypothesis of our colleague who was called Munch, or the Munch's hypothesis. Okay, so by now I think you guys have been able to understand that. Now let us think of this this question. Let us think of this question. You can study this structure here, and you see what it is showing. Now, according to Munch's, according to Munch's hypothesis, which of the following conditions, given conditions, would increase the rate of translocation? So I am giving you 1 minute to think through this question, and you probably raise your hands and you give us the answer. So you should be thinking through. We are looking at the source and then the sink. According to Munch, what do you think? And when you are choosing the objective of your choice, you should be giving us a reason or an explanation to support your answer that you have decided to choose. I'm seeing yes, like a champ.
Okay, Master, me, I think. Okay, my answer is B. Reason being increase in in the concentration of sucrose increases the rate at which the water moves osmotically into the cell, increasing its target pressure. Therefore, the water will the solution will flow through the tube faster. That's what I think.
Okay, thank you very much, Lucky.
You're welcome.
What about the rest? What do you think about Lucky's answer, or do you think her answer is the correct one, or you have a different alternative answer? What do you think? From what we have understood about Munchi's hypothesis, or you could call it the mass flow hypothesis. The mass flow hypothesis or the pressure flow. So all those names, depending, they can work. Actually, if it's a uname question, it could work all through. Okay. I need some other person to say something. Yes, Cinderella.
Yes, sir. Good morning.
Good morning, Cinderella.
Yes, I think Lucky's answer is correct. I think the option should be B.
Okay.
Yeah.
Cinderella, why not D? What makes D wrong, or what makes A wrong?
For A, when, of course, like when there is photosynthesis going on, there will be a lot of sugar conversion to, like there will be a lot of manufacturing of sucrose, which will enable it to, so when, like there will be higher concentration of sugars at the source, so than the sink, and to enable the movement of this material down.
Okay. Thank you very much. Then I need someone to tell us why they think A is, why they think D. D talks of an increase in humidity in the outside air. Why someone may think that D is wrong. I need someone to reason D out. What do you think? Anyone? Yeah, I'm seeing we are having many people here. I'm seeing we're having many people here. Rita, what do you think? Why do you think they would be wrong, Rita?
I think they would be wrong because increasing the humidity of the outside air will reduce on the photosynthesis, and there will be no, the concentration of sucrose shall be low because little sucrose will be formed and the translocation will also be low.
Okay, thank you for that. But Rita, how does the humidity lower the rate of photosynthesis? Okay, let me choose another person here. Letitia, what do you have to say? Letitia, okay.
What teacher? No.
Yes,
Teacher,
Letitia.
I think B is not correct because when there is an increase in humidity, that it reduces the rate of transpiration, which means the stomatas are closed. Thus, photosynthesis cannot take place. Yet, in Newton's hypothesis, we require photosynthesis to take place, the sucrose to be produced.
Wait, Letitia, does increase in humidity bring about closure of the stomata?
Yeah, because the water vapor on the outside is much compared to the one in the cells.
Okay. Yeah. Thank you for that. There are some people are putting up their hands. Maybe they want to say something more. Eleanor. Yes, Eleanor.
Well, teacher, for me, what I think,
Mm.
when there is increased humidity, then there is little light, therefore there is no photosynthesis, and then the sucrose which is produced is then little, so it doesn't work out.
Okay. Thank you. Then Nakanwa Jiwini, what do you have to say? Yes, Nakanwaji. Okay, Nakanwaji's her microphone is not on. Emerald.
So, okay, according to me, I think increase in humidity in the outside air is going to lead to increased water potential within the cell itself. And thus increase in water potential is going to decrease the solute potential, thus decreasing the rate of translocation.
Emerald, you are saying that humidity, that is increase, come again?
Increase in humidity in the outside air is going to, okay, the cell is going, since the cell has more solute inside, it tends to attract the water inside, thus increasing the water potential inside the cell. And the solute potential decreases. So it decreases the rate of translocation. That's what I think.
Okay, let's end with Lucky, and then we'll finalize with that. Lucky, what do you have to say?
Okay, my study is wrong. Reason being that increase in the humidity of the outside air increases the rate at which the water from the outside enters the cell by osmosis instead of the water from the xylem. Therefore, the process of translocation.
Okay, thank you very much, Lucky. Yeah, I like all your ideas, but with this point of increase in humidity, remember that water, if you can look at this picture, once target pressures are increased at the source, and then low at the sink, it is creating what we call a pressure gradient. With gradient, it means that one end has more of something and the other concentration gradient or pressure gradient or anything to do with the gradient. It means that there is more of something at one point and low of something at another, leading to that something which is more at one point to begin moving towards where it is at a low end. Get it? Now, remember that the moment fluids move downward to the sink, they're supposed to be pushed out due to building up of by hydrostatic pressures. Now remember, this water again moves to the xylem, which water is moving up the plant. And remember, one of the key reasons or factors that bring about movement of water up the plant is the transpirational pool. And once humidity is high, so the rate of transpiration is gonna be low. The rate of transpiration is gonna be low. So this water that is building up here is not moving well. So you find that the concentration gradient, or sorry, the pressure gradient is gonna be reduced in the end. And so the rate of translocation will also lower. It will also lower. So as But for C, a decrease in phloem unloading at the sink. This one obviously will just decrease, because unloading, like once here at the sink, once the sugars have come, they're supposed to be unloaded, and some are respired and then some are converted into starch. That is the unloading. So when you decrease that, it means you are not increasing translocation. Then an increase in sucrose production at the source is going to generate high tagger pressures here, which in the end may increase the rate of translocation. So this one may stand out to be correct. A decrease in photosynthesis is so wrong. Okay, I think you guys have been able to understand that question very, very well. And then we can also have this other question, which says that what mechanism explains the movement of sucrose from the source to sink? What mechanism explains the movement of sucrose from the source to sink? What mechanism explains? So you can read through those answers and you tell me. Yes, Faisal or Yang. What do you have to say?
So I think the mechanism which explains the movement of sucrose from the source to the sink would best be described by option B, because that basically describes the mass flow hypothesis, water moving into the sucrose-loaded sieve tube cells, creating a higher hydrostatic pressure causing it to move down the sink.
Onyang, your network is kind of fizzy. I don't know why it's making that, but I would love to hear your submission well, but your network, I think there has been some sounds and we didn't hear well. We didn't hear well, Onyang, but I'd love to hear. I don't know what's wrong with your network. Is there any other person with a different? It's like I had Olyang talking of a B, and that is osmotic movement of water into the sucrose loaded sieve tubes, creating a higher hydrostatic pressure in the sucrose than in the sink. That was according to Olyang, but his network has not been stable. But then we also have tension created by the differences in hydrostatic pressures in the source and sink. Then we're having active transport of sucrose to the sieve tube members, driven by the proton pump. So what about the rest? What do you think? Because for you to be able to, if you can answer some of these very well, you find that the process just becomes clear. Like whenever you find it, you will never fail to interpret anything to do with Munch's hypothesis. By the way, some people think these things, and that's why normally for most of these things, I tend to even get some UNEB questions or even different mock bodies. If you are to look through some mock bodies, this Munch's hypothesis is highly examinable. So you must understand it to the dot, so that you never get confused. So I didn't get Olyang very well because of the network, but any other person, please raise your hand and you give us your take on this question. The question still stands: what mechanism explains the movement of sucrose from the source to sink? From the source to sink. From the source, what mechanism explains the movement of sucrose from the source to sink? Yes, Lucky.
Okay, Master, even me, I think the answer is B. Like
Why?
I said, because now you see, teacher, if you see the explanation of that B answer, okay, the answer B is a real explanation of which is experiment. So like
Okay.
it really rhymes. So okay, let's add in.
Okay. Lucky, why do you think A is wrong?
Okay, A is wrong because it really has. Okay, it's okay. I don't know how to explain it, but it's really wrong.
No, you don't, in science we don't just say really or something. Okay, let's use normal Cinderella. Normal. Why do you think A is wrong?
For me, I think the option is C.
Hey.
Yes.
Can you
But
explain?
I don't have clear explanation for that.
Okay, we could go one by why do you think A is wrong? Normal, why do you think A is wrong?
Yes, I think that when evaporation takes place, water will be lost from the leaves, and that will reduce its that will reduce the water potential in the leaf, creating low turgor pressure, which will transport the sucrose from the source to the sink.
Okay. Okay. But you didn't tell us about active transport of sucrose from the sink. You didn't tell us that. Okay, but thank you for that. According to normal says that C is correct. That is tension created by differences in hydrostatic pressure in the source and sink. And I think here you must be knowing the meaning of the word tension. So when you're looking at tension created by the differences in hydrostatic pressure in the source and sink. And then for D, I don't know why people have not yet looked at D. This says active transport of sucrose through the sieve tube members driven by proton pumps. So I would like people to give me reasons, because if you can select out the best alternative basing on reasoning, then it becomes clear. Then it becomes clear. Okay, I'm seeing people maybe a little bit confused. Now, you find that when we are looking at the mass, because this thing is what mechanism explains the movement of sucrose from the source to sink. Now, when you're looking at movement from source to sink, I tell why would you begin from the sink? Because here they are looking at active transport of sucrose from the sink. So this one puts out A to be totally wrong. Totally wrong. So A is totally wrong. Then B says osmotic movement of water into the sucrose loaded sieve tubes, creating a higher hydrostatic pressure in the source than in the sink. Now, this is correct because one, this sucrose movement from the source to sink is driven by bulk flow of water. And why does the water create hydrostatic pressures at the source? It's because there are sugars that have been loaded at the source, and so water enters them by osmosis. So that is very, very correct. Then C, which talks about tension created by differences in hydrostatic pressure in the source and sink. Now this one is not so clear because with tension means it's like a force which tends to pull apart. You get it? It's like a pulling force. So this thing is not a pulling force. It's like actually a pushing force, because normally once you're having a lot of sugars being in the phloem at the source, then water entering. So it's water. It's the hydrostatic pressure is generated at the source that are going to be pushing water down the sink. You get it? We are not looking at it rather the other way around. Then the active transport of sucrose through the sieve tube. We know that movement of sucrose from the source to sink is passive according to the mass flow hypothesis. It is a passive process. So that's what makes B the most correct one. Otherwise, I think that marks the end of this hypothesis, which is Muchi's hypothesis. So the next time we shall be looking at the cytoplasmic streaming, as well as the electroosmosis and surface spreading. So for now, that marks the end. And in summary, this hypothesis was put forth by Munch, which you can call the Munch's hypothesis. And it looks at sugars being manufactured by photosynthesis, creating low water potentials at the cells at the source, which are in the leaves, and then they are being utilized at the sink, at cells in the roots. So that creates a pressure difference. Pressure gradient is generated and pushing the sugars from the source to the sink. Otherwise, is there any other question before we can end our lesson? Is there any other question concerning biology? You could raise your hand or any. Yes, Lucky.
Okay, Master, I just have a request. I wish you could send more other questions, like on the very topic, because it's really it seems confusing, but maybe by being exposed to such would be more versed with the topic.
I don't know whether you have a question bank, but I think there is a question on Munch's hypothesis in the question bank.
Okay.
Okay. Thank you for that suggestion.
Yeah.
Is there any other question concerning? Yes, Nomo, Cinderella.
Yes, sir. Mine is just like when they bring a question about the mass flow hypothesis, do we just have to explain like the concentration of the sucrose at the source and how it is transported down to the sink only?
Yeah, I don't know which other details do you want to add in?
Nothing.
Yeah, and for more of this, please go and read about this very process in a textbook. For example, when you read through functional approach, you'll find it and you will understand it in more detail, or through understanding biology by Susan Tool. Okay. Otherwise, if there is no question, we can end here. You've been a wonderful class. Bye-bye and enjoy yourselves. Goodbye.
Thank you.
Goodbye. Thanks for teaching us.
Okay, you're welcome.
Yeah, yeah.